Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on evidence from embryology. In our last lesson, we learned about the evidence of evolution from comparative anatomy. We discussed homologous structures and analogous structures. In today's lesson, we will learn how comparative embryology examines the similarities and differences in embryos to provide more evidence for evolution. Let us begin with an activity. Students, what is an embryo? Please write your answer on a piece of notebook paper.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. An embryo is a plant, animal, or human in the early stage of development. The embryo comes from a fertilized egg and grows into a fetus. The embryo contains the basic cells needed to form a mature organism. The cells contain DNA, and it is the DNA that instructs the embryo to grow. Let us watch a video about the development of embryos and what they can tell us about evolution.
Watching a fetus develop reveals much about the ancestral history of that organism. A human embryo in its third week is a featureless disc of cells until a furrow appears and plows its way across the top layer of cells. This is the primitive streak. As the primitive streak then begins to shorten, it leaves a trail in the form of a tube of tissues. This is the notochord, a stiffening rod found in embryos of all chordates. In primitive vertebrates, it persists throughout life as the main axial support of the body. But in higher vertebrates, the spinal cord replaces it. But because our developmental heritage, the spinal cord cannot develop unless the notochord secretes the right chemical signals. The genome includes the information necessary for the embryo to develop properly. But the genome has a history. It has been passed down through a chain of ancestors, unbroken since the dawn of life. And it still goes through the motions of creating many of those ancestors. 300 million years ago, our ancestors were creatures who laid their eggs in water. These eggs were small and round and contained yolk sacs to sustain the embryo. This is the first stage of a human embryo. But we notice that our embryo goes through a stage where it takes the form of a flattened disc. We can thank our reptile ancestors for this stage. When reptiles started laying their eggs with new hard shells on land, the embryos had to be supplied with a larger yolk to feed them through a long period of incubation. In order to accommodate this larger yolk, the embryo itself became a flattened disc squeezed between the yolk and the hard shell. The human embryo no longer needs a yolk sac, but even after millions of years, each human embryo is rolled out to form a germinal disc, reptile fashion, before rolling up again. There are many more times in fetal development in which evolutionary history plays a role. Human embryos all have temporary pharyngeal pouches which echo the gill pouches of our ancient fish ancestors. These pouches eventually became the structures that evolved from the gill pouches of fish. Structures that include the eustachian tube, middle ear, tonsils, parathyroid, and thymus. And of course it is not just physical structures themselves, but also the genes which control the development. Many of these genes controlling the development of human embryo are exactly the same genes that control the development of other creatures. In fact, some genes, like the Hox gene, which regulate the location and shape of limbs and parts, are able to control the development of creatures as different as flies and mammals.
Comparative embryology studies the way in which the embryos of vertebrates develop before they hatch or are born. Comparing the different embryonic stages of different animals is a tool that can be used to figure out relationships between species, providing evidence for biological evolution. The embryonic stage of many animals is very similar. For example, fish, bird, rabbit, and human embryos all look alike in the early stages. They all have gill slits, a two-chambered heart, and an embryonic tail with muscles to move it. The gill slits in the embryos are not actually gills. Rather, they connect the throat to the outside. And in many species, like humans, they close later on in development. Fish and some amphibians continue to grow these slits into real gills. Also, the embryonic tail does not develop into a tail in all species, like in humans. During the later stages of the development process, it reduces to the tailbone. Students, what do you think similarities in organisms indicate during early development of an embryo?
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. If you answered the question by saying that similarities in organisms during early development mean that they must have similar genes or share a common ancestor, then you answered correctly. As the embryos grow and develop, they become less and less similar. However, if species have a similar pattern of embryonic development, the species are assumed to be closely related. For humans and other vertebrates, although we may not look or function the same, the likeness in the pattern of development suggests that we have a common ancestor. In other words, the study of comparative embryology tries to prove that different vertebrate embryos, for example, mammals versus fish, follow a similar path of development due to their common ancestry. The developing vertebrates have similar genes, which determine the basic body plan. However, as the development process continues, distinct characteristics form in the embryo to identify the different species. For our final activity, students, can you explain why varying species embryos look different later on in development? Please take a few minutes to write down your answer.
Students, let's get ready. Begin. Time's up. let's get back to our lesson.
Welcome back, students. The answer to the activity question is that embryos look distinct from one another later on in development because unique genes in different species cause the embryo to take on different characteristics and develop unique structures. That is all for our lesson today, students. To review, we learned about the evidence for evolution from embryology. Until next time, thank you, teacher. Thank you, students.